Today we are going to learn 3D bioprinting. In today's lecture, we are going to learn about what is bioprinting, what are the various types of bioprinter, and what are the challenges of bioprinting. At the end of the lecture, we are also going to learn this bioprinting technique more in detail through practical demonstration. Let us see what is 3D bioprinting. The 3D bioprinting process involves using bio inks that contain living cells to 3D print the natural tissue-like structures. So that means we are going to use the bio ink and we are going to print a artificial organ or artificial tissue using the 3D bioprinter. And this bio ink is made up of natural or artificial biomaterials and that is blended with the living cells. And we can print the artificial organ or tissues, and that will allow the researchers to study the human body functions in vitro. So this is the conceptual bioprinter. So we can print the artificial organ or artificial tissue, and that will be very useful to understand the human body function in the lab condition. And as I told in the previous lecture also, when compared to this two-dimensional cell culture studies, we can go for these kind of three-dimensional printed tissues, so which will offer more biological relevance whenever we discover some new drugs or whenever we make new nanomaterials. We can use these kind of 3D printed tissues and we can understand the toxicity of these nanomaterials, or we can also understand the therapeutic efficiency of the drugs. Let us see the difference between 3D printing and 3D bioprinting. In 3D printing, we will be making the physical organ model which will replicate the shape and structure and mimic the mechanical properties, but there is no biological activity. Whereas in case of 3D bioprinting, so we will be making the bioactive tissue model. It will simulate like a original tissue microenvironment and imitating the original tissue structure, and it also has the biological activity. That means we will be making the tissue which will be mimic like our original tissue. So that is the 3D bioprinting, and here we will be using the bio ink which contains the cells. If you see the application of 3D printing, it is mainly useful for intraoperative navigation and also for medical education and device testing, whereas the 3D bioprinting will be useful for understanding the efficiency of the drug by screening the various drugs, and we can also make these artificial tissue for tissue regeneration and organ transplantation. From this slide, you can clearly understand the difference between 3D printing and 3D bioprinting. In the 3D printing, the materials commonly used are mainly the thermoplastics like PLA and ABS, and we can also use the metal 3D printer to print metal scaffolds which can be useful for hip or joint replacement, and ceramic printers for printing ceramic structures for bone repair and replacement. And if you see the 3D bioprinting, as I told earlier, we will be using these bio inks which is composed of living cells, hydrogels and other biomaterials to create the biological structure which mimic like your original tissue or organ. So if you see the application part, this 3D printed scaffolds are mainly useful in the manufacturing for rapid prototyping as well as for creating the complex parts, and mainly it will be useful in the automotive as well as aerospace and other industries like electronics. Whereas in case of 3D bioprinting, it is mainly focused on the medical application, including creating the tissue models for drug testing. As I told you, we can use this 3D printed tissue for screening the various drugs, and also for developing some of the complex organ structures as well as the tissues for transplantation and regenerative medicine. In case of 3D printing, the technology is already optimized, and there are variety of machines are available for different types of 3D printing. Whereas in case of 3D bioprinting, it is a more complex and it is an upcoming field because it requires sophisticated handling of living cells and also precise control of biological environments to 3D print the tissues. Let us learn about bio inks in more detail. Bio inks are used as base materials for bioprinting tissue or organ or bone structure, and selecting the right bio ink composition and density plays a very important role in the cell viability and density. So these bio inks are prepared by mixing the cells with biocompatible materials. For example, we can use this hydrogel, and selecting the suitable hydrogel or the particular biomaterial is based on the organ which you are planning to print. For example, if you want to print the bone, you can use the collagen because the collagen is widely used for bone printing, and these are the various other bio ink materials. We can use the collagen or alginate or fibrin. So these are widely used bio ink materials because it has a excellent biocompatibility, and it homogeneously incorporates the cells and growth factors, and it is very easy to process under the mild condition, and also very easy to modify by simple chemical reactions. So, and also it has a sol gel transition. So, these are the advantages of these bio ink. Let us see the difference between bio ink and biomaterial ink. In bio ink, as I told earlier, the cells are the mandatory component. So, this is a cell plus biomaterial, and by using the 3D bioprinter we can print the particular 3-dimensional tissue. Whereas in case of biomaterial ink, we are going to use the biomaterial as the ink, and we are not going to add any cells at the time of 3D printing. So, once we 3D print the particular scaffold, by layer we are making this porous scaffold. So, once you make this 3D printed scaffold, then you add the cells on the scaffold and grow the cells on the scaffold and make the final tissue. So, that is the difference between the bio ink and biomaterial ink. Let us see the advantages of 3D bioprinting. The 3D bioprinting allows mimicking the real structure of desired tissue or organ. That means we can use the 3D bioprinter and we can print the artificial organ or artificial tissue in the lab environment, and that can be useful for studying the efficiency of drugs more accurately, and again that minimizes the animal testing. So, we can study the efficiency of drugs using this 3D printed tissue, and that reduce the use of animals, and also this is a highly biocompatible and it is a automated process. So, less human errors, and again by using this 3D bioprinting technology we can create the patient specific and organ specific treatment. For example, some patient is having a tumor, right. So, we can isolate the tumor cells and grow in the lab environment. We can make a 3D printed tumor model. So, once we have this 3D printed tumor model, we can study the efficiency of different anti-cancer drugs. For example, you are having anti-cancer drug like two types A and B, and we can study the efficiency of this anti-cancer drug on this 3-dimensional tumor model, and suppose if the anti-cancer drug A is efficient, then we can give this particular drug to the particular patient. So, in this way we can improve the efficiency of the cancer therapy. We got the overall idea about the bio ink. Now, let us learn about the 3D bioprinters. These 3D bioprinters are designed to handle sensitive living cell containing materials with minimal damage, and these 3D bioprinters can be broadly divided into 3 types, that is inkjet based, extrusion based and laser assisted. Each has its own advantage and disadvantage, which I will discuss in the subsequent slides, and the compatibility between the bioprinter and bio ink is very important to get the proper 3D printed tissue. So, these are the various types of 3D bioprinters: extrusion bioprinting, inkjet bioprinting and laser assisted bioprinting. So, let us learn one by one in detail. The first one is inkjet based bioprinting. So, the principle of the inkjet based bioprinting is it uses a thermal or piezoelectric actuation to print the cells suspended in a liquid medium. In the thermal inkjet, it uses an electric current pulse to create the bubbles that propel ink droplets onto the surface. And in case of piezoelectric, so it uses acoustic waves to eject the bio ink. The size and velocity and morphology of the drops can be controlled by actuation pulses. The inkjet bioprinting is widely used for non-biological and biological application, and it is similar to your 2D ink based printing. So, most of you might have seen this inkjet printer, where you will be using this ink as well as the paper for printing. So, it is similar to that. Here, instead of the ink we are going to use the bio ink, and instead of the normal paper we are going to use the bio paper. And this bio ink is basically, as I told earlier, it is a spherical cell aggregate, it is a clump of cells, and the bio paper is a supporting biocompatible gel. So, these droplets will be printed on this bio paper, and then next layer of this bio paper will be kept, and again it will be printed. It will be a kind of layer by layer printing, and once it is printed, then we can remove this bio paper. So, The biopaper is basically made up of thermosensitive as well as pH sensitive gels. So, by simply changing the pH or by simply changing the temperature, we can remove this biopaper and allow the cells to grow until you get the final living tissue. Nowadays, we have the modern inkjet bioprinters which are designed for high precision, speed and resolution. And one of the limitation of this inkjet based bioprinting is the biomaterials, that is bio ink, should be in the liquid form for droplet formation. So, the next one is extrusion based bioprinting. So, the principle is it uses nozzle and pressure system to deposit bio ink to create these structures. And this micro extrusion bioprinter, again it can be broadly divided into three types based on pneumatic, piston or screw. And this micro extrusion bioprinter, some of the micro extrusion bioprinter have multiple print heads to disperse several materials simultaneously, and some bioprinter includes a fiber optic light source for photo initiated activation. For example, if you want to do the cross linking and based on the light, so that can be possible by using this fiber optic light. The third one is the laser assisted bioprinting. Here we will be using this laser to transfer the biomaterials onto a solid surface. So, you can see here we are going to use these laser pulses and the cells are loaded here. So, with respect to the laser pulses, the cells are deposited on the substrate. So, the substrate may be made up of biopolymers or it can be cell culture medium, and the substrate supports the cellular attachment and promotes the growth of the particular tissue. So, from this slide you can understand the difference between the various 3D bioprinters, and we have to select the right bioprinter and right bio ink depends on our final application. For example, if we select this inkjet, you can see that cell viability is up to 85% and the printer cost is low. Whereas in case of micro extrusion printer, you can see the cell viability is between 40 to 80% and the cost of the printer is medium. And in case of laser assisted 3D bioprinter, you can see that cell viability is more than 95% and the printer cost is high. So, depends on your final application and cell type and bio ink, you have to select the right 3D bioprinter. Let us learn about ideal material properties for bioprinting. The first one is printability. So, the material should be printable. So, that is depends on the viscosity, gelation methods and rheological properties of the particular material. And the material should be biocompatible in nature. That means it should not induce any adverse effects in the body. So, it should be compatible to the biological system. The next one is the degradation kinetics and byproducts. That means it should be biodegradable. So, the degradation rate should be matched to the ability of the cells to produce their own ECM. For example, you have this 3D printed scaffold, and if it is degrading, and before the particular scaffold is degrading, the cell should make its own ECM, that is the extracellular matrix, and it should be able to grow and form like a original tissue. And when the particular biomaterial is degrading, the degraded product should not induce any toxic effects. So, the degraded product should be non-toxic. So, that is why we have to check the particular material whether it is biocompatible and also it is biodegradable before we are using it for 3D printing application. The fourth one is structural and mechanical properties of the material. So, material should be selected based on the required mechanical properties. Depends on the organ or tissue which you want to print it, you have to select the correct material. For example, we can select the thermoplastic polymer fibers if you want to have a strong material, and we can use the soft hydrogel if you want to make the soft tissue. So, the soft hydrogels will provide better cell compatibility. So, it depends on your final application, you have to select the correct material with the correct mechanical properties. So, the last one is the material biomimicry. So, the main aim of this 3D bioprinting is to create a artificial tissues or organ which will mimic like your original tissues or organ. So, we have to select the material which biomimic it's which mimic like your original tissue or organ. So, we have to select the particular biomaterials. In that case, the success rate of the final product is very high. Let us see the overview of 3D bioprinting process. So, the first step is pre-bioprinting. In this step, we will be selecting the right bio ink, depends on your final application, and the second step is the processing. So, here we will be selecting the suitable 3D bioprinter and print the particular tissue. And third one is once you printed the tissue, in the third step, that is a post bioprinting, where you will be stabilizing the bioprinted construct with cross linking, And we can also do the further modification, like we can remove the sacrificial inks and seed with the cells. So, the sacrificial ink means it is mainly used for creating the channels, open channels, which is essential for nutrient transport and cell migration, and in some cases it can be useful for vascular network design. And these sacrificial inks can be removed simply by dissolving the water, or it can be degraded at specific temperature. By simply changing the temperature, we can remove the sacrificial ink. Let us learn about what are the challenges we face when we do the 3D bioprinting and how to overcome those challenges. So, the first problem is poor bioink formulation. The reason may be due to the polymeric solution flows freely. So, that can be overcome by measuring the viscosity before loading the cells to ensure the injectability. And the second reason may be due to the cells are not viable in the matrix. So, in this case, we have to check the biopolymer cytotoxicity before we loading this biopolymer into the 3D bioprinter. And third reason may be difficulty in post bioprinting process. So, that can be overcome by using the proper cross-linking agents and appropriate bioprinter. So, the next problem is polymeric clogs in the bioprinter. So, that may be due to the improper bioink formulation. So, in this case, we have to check the polymeric properties like mechanical strength, rheology before we use it for 3D bioprinting. And the other reason, it may be due to the misaligned bioprinter axis and parameters. In this case, we have to contact the technical scientist for calibration, and the speed may be too low or fast during the process. So, we have to optimize the parameters, and you have to standardize the bioink before we take it for 3D bioprinter. So, the next problem is deformation post printing. So, the reason may be due to the low cross linking density. So, you have to optimize the curing condition or cross linking parameters, and another reason is high ambient temperature or humidity. So, we have to optimize the temperature and we have to use the dehumidifier, and cells are not viable after 3D printing. So, the reason may be due to the cross linker toxicity. So, we have to check the toxicity of cross linker by using the viability assays, or it may be due to the improper process. So, analyze the porosity, nutrient absorption and metabolite release study whether the cells are viable after optimizing all these parameters. Let us see the role of nanomaterials in bioprinting. The traditional bioink has some limitations. For example, so the traditional bioink face challenges in rheological, mechanical, physiochemical and biological properties. So, this traditional bioink can be improved by use of nanomaterials. And when we are using these nanomaterials, and it can improve the efficiency of bioink, and it can mimic like your natural tissue features. So, in this way we can improve the bioink and with the enhanced properties for the 3D bioprinting application. So, when compared to the traditional bioink, this nano bioink has a lot of advantages. So, it has the nanomaterial, it has the cells as well as it has the biomaterial. So, this nano bioink can be useful for various applications. For example, if you are using this magnetic nanoparticle in the bioink, then we can, with the help of external magnetic field, we can allow the drug to release in a slow and sustained way. And we can also have a photoresponsive materials, we can add it. With the help of light, we can control the release of drug or control the release of growth factors. And we can also have the pH responsive materials, as well as we can also have some of the nanofibers as well as some of the nanosilicates. So, that can improve the mechanical strength of the particular bioink. Let us see the role of nanomaterials in 3D printing. So, here we can use the nanomaterials like cellular nanoparticle, which has the antibacterial property, and we can also use these nanoliposomes, which can be loaded with the anti-cancer drug. And these nanomaterials can be loaded into the 3D printed scaffold. And once you remove this bone tumor, and there will be a defect. So, that defect can be repaired by using this scaffold. This scaffold is already loaded with the anti-cancer drug. So, it will kill the residual cancer cells. At the same time, it can also support the bone regeneration. Let us see another example to understand the role of nanomaterials in bioprinting. So, whenever we print the cardiac tissues, so we have to make sure that the printed tissue is able to sustain electrically excitable cardiomyocytes and exhibit robust and synchronized contraction for functional 3D cardiac structures. But the challenge with the current bioink is most of the cardiovascular bioink is made up of polysaccharide or protein polymers, and it lacks conductivity, so which hinders the electrical coupling with the host myocardium. So, to overcome that, we have to incorporate conductive nanobiomaterials like gold nanorods into the bioink, which will enhance the electrical impulse transmission and improve the cardiac cell adhesion organization, as well as it will provide the synchronized contraction. As I told earlier, we can also use the 3D printed matrix model for assessment of nanoparticle toxicity. So, when compared to the two-dimensional cell culture plate, we can use this 3D printed three-dimensional tissue for understanding the toxicity of nanomaterials, because this three-dimensional bioprinted matrix model, so that closely mimic the real tissue environments, and it will provide a more realistic assessment of nanoparticle toxicity. Let us briefly learn about higher dimensional cell culture model. So, the first one is 4-dimensional scaffold. So, this 4-dimensional scaffold responds to the external stimuli. So, once we do the 3D printing and loaded with some kind of magnetic nanoparticle, with the help of external magnetic field, the particle scaffold can shrink or expand, or if you are adding some nanoparticle which can respond to the light, so the scaffold can shrink or expand with respect to the light. So, that means the 4-dimensional scaffold is basically a 3D scaffold, but it responds to the external stimuli. Responds to the external stimuli, that is called 4-dimensional scaffold. So, the next one is 5-dimensional scaffold. So, this 5-dimensional scaffold involves rotation motion of the printing head at defined angles, especially the curved layers. So, you can see here it can rotate in all the direction. So, that is 5-dimensional scaffold. The next one is 6-dimensional scaffold. So, it is a combination of 4D plus 5D. So, it responds to the external stimuli, and it also has a 5 degrees of freedom. It can also rotate. So, these are the difference between the 4D, 5D and 6D scaffold. I hope you got the overall idea about the 3D bioprinting. Let us go to the lab and learn this technique more in detail. Before proceeding with the experiment, we will UV sterilize the biosafety cabinet and all the required materials. In today's experiment, we will perform 3D bioprinting using GELMA as a biopolymer, which should be synthesized by the gelatin with the methacrylic anhydride. We will begin by preparing a 10% GELMA bioink. To do this, we have already weighed 1 gram of GELMA, which will be mixed with 0.5% LAP, that is lithium phenyl 2,4,6 trimethyl benzoyl phosphonate, a photo initiator. Now, we will add 10 ml of this solution to the GELMA. Once the solution has been added, we will place the mixture at 37 degrees Celsius until the biopolymer is completely dissolved in the photo initiator solution. Then place the solution in a dry bath at 37 degrees Celsius until it is fully dissolved. After some time, you will observe that the solution is completely dissolved. Now, we need to load a sterile syringe with the GELMA solution. We will pour the solution into the syringe and carefully push the plunger up until a meniscus forms at the tip of the syringe. The plunger will also help control the flow and prevent dripping. There are two extruders available. We can use either one, but the extruder we select later must be connected to UV cross-linking light. For now, we will place the syringe into extruder 1. Once done, we will begin the bioprinting process. First, we need to switch on the machine so that it can align to its ideal position. Then open the software on the PC and connect to the machines through Wi-Fi. Log into the software through Chrome by entering your credentials. Once you logged in, the available bioprinting machines will be displayed. Select the correct machine and press connect to establish a connection. The machine will then be ready to operate through the PC. If the machine is idle, meaning there are no ongoing projects, we can begin setting it up for bioprinting. First, upload the STL file of the design you want to print. Next, specify the layer height and print speed, which will define the height of each layer and the speed at which the machine will print. Then choose the build plate. In this case, we'll use a cell culture dish. Next, calibrate both extruders, even though extruder 1 already contains a sample. Start by calibrating extruder 1. If you press the X-axis calibrate button, extruder 1 will move to its origin position in the X direction. Press the Y-axis calibrate button to move extruder 2 to its origin position in the Y direction. For the Z direction, we must be very careful as it defines the distance between the needle and the surface where the design will be printed. We need to leave the minimal distance according to our layer height and the needle type. Press the button to calibrate. Once extruder 1 is calibrated, press the up button on the screen to calibrate extruder 2. This extruder will be used for UV cross-linking, so calibrate it accordingly. Next, we need to set the parameters for extruder 1 such as temperature and pressure, which should be adjusted according to the biopolymer we are using. For UV cross-linking, the settings will vary based on the polymer's cross-linking ability. For extruder 2, its only role is UV cross-linking. So no other settings are required for printing. Once all the settings are configured, press print. The machine will start the bioprinting process. Extruder 1 will begin printing the design layer by layer. After each layer is completed, extruder 2 will apply UV cross-linking for 60 seconds to cure the layer. This process will alternate between printing and curing after each layer. Once the printing process is complete, you will see the printed design on the build plate. In this demonstration, we have shown the basic steps involved in bioprinting, including the necessary parameters for using Gelma as a biopolymeric ink. These steps are essential for successfully carrying out a 3D bioprinting process. As a summary, in today's lecture, we learned about what is bioprinting and what are the various types of bioprinter, and also what are the challenges of bioprinting. And through practical demonstration, we also learned this bioprinting technique in more detail. Thank you for your kind attention. I will see you in another interesting lecture.